This morning, my former advisor, Jennifer Doudna, and her longtime collaborator, Emmanuel Charpentier, shared the Nobel Prize in Chemistry for their discovery of a new genome editing tool called Crispr. When I was a PhD student here at MSU working in the joint laboratories of Mark Young and Trevor Douglas, we were studying the viruses that live and thrive in the boiling acid hot springs in Yellowstone National Park. But what we found about those microorganisms that are living there is that they're constantly being infected or preyed upon by viral predators. At the end of my PhD here at MSU, I was interested in viruses and bacteria and how bacteria fight off these viral infections. And Jennifer Doudna's lab was experienced in structural biology, and they were studying a phenomenon that was really hot in science at the time called RNA interference. Our idea together was to work on CRISPRs to understand how these CRISPR systems work, and in fact, if they rely on a small RNA-guided mechanism to fight off these viral infections. CRISPR is an easy way to program a molecular scalpel to find a piece of DNA and cut it. And by cutting it, that's the first step to surgery, to repairing genetic disease. CRISPRs have proven to be remarkably versatile. So not only can they be used for different aspects of genome editing or engineering in a wide variety of different ways, they've also been repurposed for diagnostics, which is particularly relevant in the current pandemic. So several new CRISPR tools have recently been developed to quickly identify SARS coronaviruses, and they're being implemented in clinical diagnostics, and my lab is working on a new version of that right now. CRISPRs have democratized genome engineering, meaning one of the reasons that CRISPR has made so much noise is that it's so easy. Before, it would have taken a room full of PhDs and a decade to engineer a single cleavage site in the human genome. Now that can be done in an afternoon by a high school student. Many labs, and all over MSU, but all over the globe, are using these now as routine molecular tools in their research to study cancer, any sort of disease, or any aspect of biology for that matter. I hope the success of CRISPR really shines a spotlight on the importance of academic research. Nobody went looking for a genome engineering tool. It was something that we stumbled on by looking under a totally different lamppost. And it turns out that under that lamppost was the answers to genome engineering in ways that we could have never anticipated. And that's sort of the perennial outcome of basic research science, is that there's these byproducts that are innovation, ways to use findings that could have never been predicted that lead to disruptive technologies like CRISPR.